we get experimental science. We're curious, non-judgmental.
Thomas, take it away, my friend.
So, maybe at this point we could talk about the pieces that we're going to do. So we're going to extract DNA. And the reason we're going to extract DNA is the informational molecule of life, and it's essentially the the informational molecule of almost all life on the planet. And there is never an absolute in biology. Valent, as you well know, you can pretty much never say never. So, what's another molecule? Well, we just went through the plague based on a maybe organism, depending on how you define it, where the informational molecule is RNA. But most of the time, it's DNA. And so in my friend, the banana, the informational molecule that makes a banana, not a strawberry, is in fact DNA. The molecule that makes me human, that makes my kid look like me, and much to my ex-wife's chagrin, sound like me and act like me, that's DNA. So it's the same molecule, and it's amazingly the same molecule. The chemistry, the biochemistry, the structure of the molecule is entirely the same across all organisms on the planet. And we're going to extract it from both of us. So I've got the pieces. We can run through the pieces. So you need a sample. Sample is banana or spit. Okay, so we're going to collect saliva. So if you're collecting saliva at home, you need a shot glass. I did actually, because I'm home, I grabbed a shot glass and I was like, oh, yeah, I could do this. If you have one, by all means. And nothing that we're going to do is hazardous. Like this protocol, I have seen it written up as you can drink the shot at the end of the day. I would strongly recommend you do not at any case, but I'm going to use a disposable shot glass.
And one feature of that, right, is because it was for kids too. So like safety concerns, like whatever you're doing in the classroom, that way it's safe for kids.
Yeah, I would say that it's never safe to drink a biological experiment, whether you're 55 years old or 6. But yeah, we do need to be careful when we're working with kids. And I don't do the spit thing with kids because I don't want them to decide, hey, maybe I'll give that a taste. Okay, so we're going to need a biological sample. So it's going to be either your saliva or a banana. The DNA is in the cells. We mentioned the size of the DNA. DNA is so incredibly thin that you cannot resolve it with a light microscope. You can get crystals of DNA where you actually get DNA of high enough concentration and low enough water that it crystallizes. And those crystals will diffract X-rays. But just try to imagine how small something has to be to the point where you can't even resolve it with an electron microscope. And the only thing you can get it to do is bend the path of X-rays. Okay, but it's in the cells. It's also incredibly long. So the DNA in every one of our cells is about 2 meters. In American, that's really long. In American, 2 meters works out to just over 6 feet long. So you've got in every cell, and you've got in your body millions of cells. You've got 2 meters of DNA, 6 feet plus of DNA. This is thread that is 6 feet long, but so incredibly thin that you can't resolve it with a microscope. It's trapped, it's compacted, it's held in a cell. It's the information of the cell. Information has to be accessible. And so there's this huge sort of battle between getting this really big, really long, really thin, really long molecule into a really tiny cell, but doing it in such a way that you can actually still access it. So think about taking a book and cramming that book down really small. You can't read the book. So the cells have evolved over time that this DNA molecule is really compacted, but at the same time also selectively accessible so that you can read the right chapter, the right sentence, the right paragraph in that book. We're going to work with that packaging. So we're going to start by physically breaking up the sample. If we're spitting in a cell into a shot glass, you don't actually have to do that. But with the fruit, we're going to break it up. And then, actually, so I'm going to tell you what the ingredients are just to make sure everybody's got the ingredients. And then we'll go through what each ingredient does as we go through the protocol. So awesome. You need sample. And we'll tell you why later. You need some kind of just soap. You need a little bit of salt. This is literally the salt out of my cupboard. If you're going to use fruit, we're going to want to separate the physical parts of that fruit from the solution that has the DNA in it. And so we're going to do that by filtering it through my friend, the coffee filter. And so you'll need a coffee filter, something to catch it. In this case, it's just a cup, and then a rubber band to hold that coffee filter in place. And then we're going to use a little bit of contact lens solution. So it ends up within the cell. There's a lot of DNA. There's also a lot of proteins. And so we can get a little better extraction if we break those proteins down. And it ends up that one of the things that contact lens solution does is it breaks down proteins. So the reason it cleans your contacts is it has an enzyme called proteinase that breaks down proteins. And so we're going to add a little bit of that to our mix to try to break down the proteins and get a little cleaner DNA extraction. So we've got sample. If you're using fruit, you're going to need a little bit of water, filter, salt, soap, and then alcohol. So you can do two different things. This is high-proof isopropanol. So isopropanol comes in a number of different percentages. This is 99% isopropanol. I think this actually works with 70%, but I don't remember if I've tried it. The other thing you can do if you're doing this at home or by any chance in a bar is you can use overproofed booze. So I have a bottle of Polish vodka in the freezer that's overproofed, and this is 70%. If you're using overproofed, Everclear is one of the American equivalents, this works better if it's chilled. Do not take shots for two reasons. One, it's a biological experiment, and B, you never want to take shots from high-proof alcohol. Ask me how I know. Yeah, so it's normally your typical alcohol is about 40%, and so if you're taking a shot that's 40% alcohol. If you inadvertently or at the egging on of your graduate students take a shot that's 70% alcohol, it's incredibly harsh. Okay,
I like in chat our friend Tommy. He's like, Thomas, I have to work tomorrow.
yeah. Anyway, that was a one-time experience, and we haven't repeated that. Okay, so I think that's all of the pieces. Do you want to start?
Yeah, let me also grab a shot glass. I forgot a shot glass. I will be right back and grab that. Otherwise, I'm ready to go. I've got my banana and everything's all set. I just need that final element to it. So let me go ahead and
while you're grabbing that, I'm going to chat. Perfect. So if you're doing the shot, you want to start now because it's going to take a little bit to get enough saliva to do this. We're going to take a piece of banana that's about somewhere around 3/4, half an inch. Do not do the classic American bigger is better. If you put too much banana, you get enough pectin that it ends up gelling at the end, and we don't want that. So we want a chunk of banana that's maybe something like the tip of my thumb kind of thing. But you're going to need that much saliva to make this work. And so you need to start spitting in your shot glass. I mean, it's never pretty. You just keep going. And you're like, wow, that's not a lot of spit. It's not. And you got to just keep going. And keep spitting in that shot glass. I normally teach in the morning, and so I often get a classroom full of kids that have just come from the Tim Hortons, and so we get a bunch of coffee-colored saliva. It's not awesome. But if you're doing the saliva thing, just as we're going through these steps, keep getting that spit together. And you want to slosh it around your mouth and get it as biological as possible, because what you're doing is picking up the cells in your mouth off of the inside of your cheek, in the saliva itself, and what we're going to do is extract the DNA from those cells. So all I was saying is if you've got your shot glass, start putting the time in with filling this thing, because you're going to want at least half an inch of spit to make this thing work.
Lovely, right? Some good ASMR for y'all chat today.
Yeah, and this is it there's no way around it. You just that would be my dog Tiny saying hi to my girlfriend Tammy.
Hi.
And that would be Tammy saying hi.
Hi, Tammy.
Maria says hi.
Hi.
Okay, so we're going to keep working on the saliva. If you're working at home on your banana, overripe banana actually works really well because it breaks up really easily. I'm going to use just the banana that I picked up at a supermarket this afternoon. It's not quite as ideal, but it'll break down. So I'm going to take a piece you're going to want to take the skin off of the banana, and you want to chunk something like this. And you can totally do this with a little bit more, a little bit less. This is not rocket science. I would cut that in half. So the issue is if you use a lot of banana, you get a lot of pectin, and that's perfect. And it's going to end up with jelly. So we're going to take the banana, and we're going to put that in a Ziploc. And we're going to add to that Ziploc about as much water as you have banana. And this is literally as scientific. Whoops. Okay, that's a little bit much water. So this is not an exact science, but you want something that looks. Yeah. So let's go, and then we'll see what happens. We may end up coming back. It ends up a little bit more water is fine, because it just makes it a little easier to pass through.
And we had a question, Thomas, about how much is too much onion.
Okay, so onions, I don't know. I think the default is going to be use about as much water as you have onion. You're going to find that onions are really tough. And so what we're going to do is the DNA is inside the cells. And if you want to picture, you've got this piece of fruit, this piece of onion. We've done this with grapes. We've done this. I started to say with my kids, like a way to keep Lila engaged was find anything in the house that you can extract. And it ends up that different fruits, you get different amounts of DNA, and for a lot of different reasons. But one of it can simply be that it's harder to break down some cells than others, and the DNA that we're looking for is inside these cells. So we're going to take this and just mash it. Okay, so Blunt, you and I are going to mash these, but we can't ignore our saliva, and we got to keep spitting in this cup as we go. Okay, so whatever fruit you're working with, whatever vegetable you're working with, we're going to mash this up, and we want to get it to the point where all of the banana is basically disappearing into this solution. And so I'm getting there, but I'm not quite there yet.
And then Thomas, we were question wondering today about what is the diploidy of the fruit?
That's awesome. Okay, so let me answer that in a two-step process. Our answers. So one, what we're doing here is we're breaking down the physical structure of the fruit to get to the point where we've got individual cells. Inside every cell is the genome, the DNA of the organism. One of the reasons that different fruit gives us different amounts of DNA is a thing called diploidy. So you and I are diploid. We've got 2 copies of every chromosome. The fruit flies that I work on are diploid. They've got 2 copies of every chromosome. A lot of plants are also diploid. In general, the idea of diploid is you get 1 copy from each parent. You've got 2 parents. A lot of domesticated plants are not diploid. They have more than 2 copies of the genome. And so bananas are actually, there are lots of different bananas, and different bananas have different ploidy. If you have this banana, it's a Cavendish. It's the same, it's actually a clone. It's exactly the same banana the entire globe over, and it's a triploid. And there's a really good chance that this banana won't exist in 50 years because there's a fungal parasite that's killing off bananas. And 100 years ago, the banana was a Gros Michel, and it was actually wiped out by these very similar fungus that wiped those out. So these bananas are triploid, and that means that they've got 3 copies instead of 2 copies, and that actually makes it a little bit easier to extract the DNA. There's a little bit more DNA in the cells. Okay, so if you've got
more questions coming in, Thomas. Is this like hydrolysis of the step that we're doing?
Sorry, I missed that one. No, not exactly. So hydrolysis would be using water to, hydrolysis would be using water to break down bonds. There probably is some hydrolysis going on here, but generally we think at this stage that we're just physically breaking up the cell wall and the physical structures of the banana. It's a really interesting question. I don't think you could say there's not hydrolysis, but we're not really driving a hydrolysis reaction. Okay, so we're going to keep spinning, and we've got our bananas. So you're going to take, zip the Ziploc for a second, set it aside. We're going to take our plastic cup and our coffee filter. It's a little too late to point this out, but in general, the cheaper the coffee filters, the better this works. So I have really good coffee filters that I use for making coffee.
Tom. Yes.
No, I was just asking, you got sodium chloride?
Yep, I got it.
Because we have the regular salt we have.
I got this salt.
That's mostly potassium.
Is this potassium salt work, Thomas?
I don't know. It should. If it doesn't work, we'll say it's potassium.
All right, there we go.
Well, it's like 50% potassium, 50%
It'll work.
sodium.
Okay.
It'll work. This thing is so robust, you can almost forget the salt and you're fine.
Thank you.
All right.
And we'll get to what the salt's going to do in a moment. Okay, so you take your coffee filter, the cheaper the better, push it down into your cup like this. We're just making a little funnel, and then fold it over, and then we're going to hold it in place with our trusty rubber band. And now you've got a filter. Okay, so with the saliva, the cells are in there, and there's not a lot else. There's not much physical structure. That looks great. With the banana, we've got a lot going on here. We've got water, we've got fruit, we've got a lot of cellulose, we've got a lot of cell walls, and we want to pull that solid stuff out. I mean, you can see how viscous this is. That's going to stay there. And so we're going to take that and we're going to filter it out using our filter. And so we're going to take our Ziploc, open it up, and reasonably carefully pour it into place. And you want to get as much of that out, but again, this is all an approximation. But you can see, kind of, it's starting, there you go. You can see it dripping through. And so we're going to let that go for a minute and just use gravity feed. Brilliant. So you can never use this glass again. Totally kidding, you're fine. You're absolutely fine. Give it a quick rinse and you're good to go. Okay, so we're going to use gravity to separate out the solids from the liquid and maybe give it a little swirl, try to help it along.
So one catch is, right, we're not going to get all the cells from this because we're not able to mush enough.
Yeah, and so when we were writing the paper, we were trying to put in things of what are additional steps that you could do? If you can do this with preschool students, are there things that you could do with a university class to make it more scientific, to a little bit more rigorous? And so one of the things that we did was, can you quantify the amount of DNA? We're not saying that you can weigh out a piece of banana and say, okay, we're going to get X grams of DNA. But you can actually use if you're in a research lab or a teaching lab, you can use a spectrophotometer and a given wavelength of light to tell relatively how much protein to DNA you have in the sample. And so we suggested this as sort of a next step. So we're not saying that you're going to do a quantitative extraction, but you can qualitatively say, how clean is my extraction by looking at that ratio of the absorbance at the wavelength that DNA absorbs light versus the wavelength that our protein absorbs light. All right, so I've got that's enough liquid that I can make this work, but let's take it up a notch and see if we can do this. Okay, so all we're doing at this point is filtering out the solid. And so we're doing this with gravity, but you can help things along. You want to do it gently, and this is something we don't do when we're doing this with kids, because trying to convince a 10-year-old or even an 18-year-old to gently squeeze the coffee filter doesn't work. But if you take off your rubber band and hold everything upright, you can get your sample basically like this, and you can just give it a gentle squeeze to try to help things along here and recover a little bit more liquid. What you don't want to do is burst your coffee filter and spray banana guts into your Dixie cup. If you do, you just start over. It's not the end of the world. Just run it through another coffee filter. So I'm going to give this a couple of more squeezes.
Cliff says, I know my coffee filters. If I squeeze, it'll break.
Yeah,
Sparky says...
So don't squeeze. This is much better to leave that. I'm going to grab a paper towel because I got banana fingers. Okay. So
Thomas, we have to have the banana animation.
Very nice. Very nice. Okay. So you have something that looks like this, whether it's an onion or a banana or a strawberry. You've got I've got a little bit less than half an inch of water, and my spit is almost exactly the same height. It's pretty frothy, which is not ideal. It's also not the end of the world. Okay, so we've now got the DNA in the cells in the solution, either here or here, and we want to get the DNA out. And ultimately what we want to have happen is we're going to get these really long, incredibly thin threads. We're going to get literally thousands and thousands and thousands of copies of these released at the same time. One of the characteristics of DNA is the backbone that makes up DNA has a slight negative charge. And so if you have thousands of copies of this string, this thread, that are all negatively charged, they're going to push each other apart. And what we're trying to do is get them to clump. And so to allow them to clump, which is going to be sort of the last thing that we're going to do, is we're going to try to neutralize that charge. And so we're going to simply add a little bit of sodium chloride, which is just table salt, and the potassium chloride should work fine. And we're going to add a little bit. And a little bit is literally the scientific measurement of take about a pinch and add it to each of those. So we're going to add a pinch of salt to both cases, and that salt is going to dissolve. We're going to gently swirl the solution in both cases.
So Wombat was wondering, Thomas, at this stage, is the DNA still in the cell, in the seep, or where is it?
Yeah, so I was just going to get to that. It's a really awesome question, Wombat. So what we're trying to do is get the system ready so that when the DNA comes out of the cells, it's at a point where we're going to allow it to stick together. So right now, the DNA is still compacted in the cells, in fact, compacted in the nucleus, but we've now created the solution that when we release that DNA, that sodium is going to be present to coat those threads. Okay. So the next step in releasing that DNA, we physically mash this up. We've spit in our shot glass. We've got the DNA in these cells. The cells in animal cells, like in our saliva, but also plant cells in our banana, have a membrane that surrounds the cell, and then inside that another membrane that surrounds the nucleus, and inside that is the DNA. So we need to break those membranes, and we're going to do that by adding dish soap. Before we do that, we're going to go back to the idea behind having this kind of a protocol. So as I'm doing this, I have literally done this extraction 100 times. And to say that I can do it in my sleep isn't entirely fair. I am sure that I have dreamed doing this in one way or another. So we're just following the protocol through here. One of the things that we did in publishing this paper was say, okay, when you get to this step, you can go, so you've added the salt, we're going to add an enzyme, which we're going to do in a second. But you can also not add the enzyme. And so one of the things you can do with this as a teaching tool is say, okay, what happens if you do this or if you don't do that? Okay, so we've added cells, our sample, we've got water, either in the saliva or that we've added to the banana. We've added a little bit of salt. The next step is we're going to add a little bit of our contact lens solution. And so
before we add that, Thomas, we have a question from the chat. Is there a way to speed up the filtering process without squeezing?
So you can add a little bit more water. It ends up that we have a ton of DNA in most samples. And so in general, what you want to do is get a solution that's at least about half an inch, just so that it's deep enough to get that extraction. If it's going really slowly, add a little bit more water and give it a gentle swirl to try to get that going. And then just run with it. And if you've got about half an inch, maybe I'm probably closer to a quarter inch of solution, you're good. You'll be fine.
And Thomas, we are linking, just so you know, your link tree and your lab Instagram is a rotating command that's coming. And for anyone wondering, the paper is posted on the Discord, the protocol, 2 places, the general announcements and mentioned on stream. So you can go ahead and follow along if you missed it today, or we'll also link it in the episode description later. Sam was wondering, Thomas, so pinch of salt, what is that relative to in terms of the liquid amount, like to getting that ratio right?
Yeah, and so it ends up, it's almost impossible to get the ratio wrong. And so I literally, I put some in my palm, I pinched it up, I dropped it in there, and then I did that again. If I had twice as much liquid, I would put that same amount of salt there. Okay. There's a lot of salt in a pinch of salt. And as long as it completely dissolves, we're good to go.
Oh, and okay. Thomas, the paper is also in the link tree, I guess. So the chat, the link tree link that is pinned to the top of the chat, you can also click there and that'll take you to the publication as well.
Awesome. Okay, so I mentioned in passing that the DNA, so chromosomes hold, I mean, how to phrase this? Chromosomes are the physical structures that DNA are part of. And if you picture a cell, oftentimes what we picture is that little circular cell wall, maybe a nucleus, and then we have these little X-like things in the nucleus. Those are the chromosomes. I can tell you that chromosomes almost never look like that, except when they're being really photogenic during cell division, and they're easy to picture, or easy to take a picture of. But those chromosomes are made up of DNA and also 50% protein. It's almost an even match of protein and DNA. We want the DNA, we don't want the protein, and so we're going to take our contact lens solution that has enzymes in it that are proteinases, enzymes that break down proteins, and we're going to add somewhere between 6 and 120 drops of protein solution. So, like 10 or 12 drops, and you're good to go. And again, if you've got twice as much solution, use 12 drops instead of 6, or use 20 drops, whatever it is. Okay.
And we had a question actually about this, Thomas, before stream today. Does this mean that you shouldn't drink it? Like, because we've seen, like, in pop culture and whatnot, that by having contact lens fluid, you might throw up or get really sick. Like,
okay, so the default is you should not drink this. We're about to put dish soap in this, and I can tell you, luckily not from personal experience, that you really don't want to drink dish soap. I would say that it is absolutely fine to drink contact lens solution, but I wouldn't ever recommend it. So, I mean, you're going to put it in your eyes. I mean, you're not going to squirt it in your eyes, but you're going to end up with some in your eyes. Don't drink this. But don't drink it just in general, not simply because it has contact lens solution in it.
And then a follow-up question, is this nuclear or mitochondrial or both?
Yeah, that's a great question. So the backstory on that is most of our DNA is in the nucleus. There's a separate genome that's actually bacterial-derived that's in the mitochondria. This is both a combination of nuclear and mitochondrial. Because both of those, the nucleus and the mitochondria, are surrounded by membranes that we're going to break down with the dish soap. The ratio, though, is such that there's so much more nuclear DNA that we're probably not seeing an appreciable amount of mitochondrial DNA. But it should be all extracted out at the same time. Like if we use this as a sample, you should be able to find mitochondrial DNA.
Chat was wondering, too, how would you separate them?
Magic.
All right.
So actually, the way that you separate them is almost freaking magic. I've never separated them. I've always done extractions. So when we do professional extractions in the lab, we do essentially a commercial version of the extraction here, and that extraction pulls out both mitochondrial and nuclear DNA. at a time when we couldn't use molecular biology to separate those, we physically would want to separate mitochondrial from nuclear DNA, and we did it with a density gradient. And so you can actually take cells and run them through you basically put them into a saturated solution of cesium chloride and spin it so incredibly quickly that tube of cesium chloride goes from being really high density to low density at the top, and the mitochondria migrate down, and you can actually pull them out with a syringe. So we use density centrifugation to physically separate out mitochondria. In the modern molecular world, we don't usually have to do that because there are other techniques that you can use to pull that out. So you can do it. It just takes a $50,000 centrifuge and a bunch of tubes of cesium chloride to make it happen.
For the soap, Thomas, we have a question. Is lemon scented okay to use?
Never use lemon scented. Yeah, perfectly fine. Absolutely good. Again, I wouldn't drink it because it smells like lemons. I've got Dawn, which this is original scent. Yeah, see, there you go. Oh, wow. Okay, I don't know what that is, but yeah, the scented won't matter. The DNA at this stage is still in the membranes, so it's in the nucleus, also in the mitochondria. But we've added the proteinase so that enzyme is there when we release it, and we've added salt so the salt's there when we release it. The next step is we are going to add, and this is more or less important. We're going to use usually 3 or 5 drops of dish soap. Let me walk you through this before you do it. Once we add the soap, the membranes that are holding those incredibly long DNA molecules are going to start to lyse. They've started to lyse a little bit just because of the physical stuff we've been doing here, but they're going to really lyse when we add the detergent. That detergent breaks down the membranes. The membranes are made of fats, they're made of lipids, and detergents as a molecule are designed to break down lipids, right? So as soon as we add this detergent to the DNA, that detergent's going to break down the lipid membrane, and that DNA is going to start to release. At this stage, we can start to shear the DNA if we're not careful.
So what is it that we need to do to be careful to avoid shearing?
So what we're going to do is add those 3 to 6 drops of detergent, and then leaving it on the tabletop, we're going to swirl the solution to mix that detergent.
So this time it's not the more intense swirling that we've been doing, but like much lighter.
So I can do this, okay, because the cells are still intact. Once the solution has the detergent in it, I'm going to leave it on the benchtop or the cutting board, and I'm going to do this to just more gently swirl that. Oftentimes we get more or less extraction, and I think this is the stage. And it's one of those things that, like, you can find the hyperkinetic kid in the group, and he tends to get sort of not great extraction because he's really spinning that, swirling that. So let's go ahead and add, whoa, that's exactly 6 drops of solution of detergent to both of those.
Both of them, right? It doesn't matter if it's the spit or the...
That's right. Okay. Yeah. Okay, so we've now got these solutions and we're going to mix them. One of the reasons that I like using the colored detergent is you can actually see if it mixes. So if you've got clear detergent, it's fine, it's great. It's actually what I use in the kitchen. But if you have something that's blue or yellow, when you mix that into your sample, you can see it diffusing through the whole sample, and you know that you've got it mixed. All right. So we are now ready for the last step. The last thing, I actually forgot to mention this, you're going to need a stick to pick up your DNA. So I've got 2 samples, so I'm going to take 2 sticks. And the shot glass one, basically all the pieces that we've done here you can find in your kitchen. The one with the shot glass is designed that you could literally be sitting in a bar somewhere and do this extraction. So I'm going to follow that sort of theme, and I'm going to use isopropanol, the rubbing alcohol, with the banana, and then I'm going to grab that ice cold overproof vodka out of the freezer, and we're going to do that secondarily with the spit. Okay, so the trick here, and again, let me walk you through it. We're going to take our sample, we're going to put it on an angle, and we're going to pour the alcohol slowly down the side. What's going to happen is alcohol and water are two liquids. They have slightly different properties. It's not exactly polarity, but it's related to polarity. It's called the dielectric constant. That difference in dielectric constant between the alcohol, between the alcohol and the water, that difference is going to mean that the DNA that is happily in solution in the water will come out of solution at the interface between water and alcohol. So we're going to see a cloud of these strands of DNA precipitating out of the solution at that interface between the water and the alcohol. So you want to pour that like you're doing a fancy shot, and it's going to layer right across the top. And then we're going to do a gentle swirl to get a little bit of mixing. And what we should see pretty quickly is the DNA coming out of solution.
So is the swirling that we do also, we should keep it on the tabletop?
Yeah. At this stage, we're going to do everything really gently.
And then what happens if we shear the DNA? Like, is that just going to be still strands, but a lot of broken ones, or what is going to be the appearance of that?
Yeah, so it should still extract, and you'll get a nice little cloud going on. But what's going to happen is when we run our coffee stick, little stir stick, through that, instead of clumping, it's sort of going to part the waters on either side.
Uh-huh.
And so what we're looking for are these incredibly long strands that have clumped together, and they're going to be easy to scoop up. If we've sheared the DNA, you're going to have these incredibly long strands that are broken into incredibly small strands. And so you're going to get sort of a scrambled egg kind of thing. We'll see. So usually, even if it's a little bit over-sheared, you'll still get quite a bit of DNA that you can see, and then still you're able to pick up some of that. So I'm going to go ahead and pour the isopropanol. And so the amount is basically dependent on how much water you've got there. So I'm going to pour about the same amount of isopropanol as I have water. Yep. There you go. And I can, as I'm looking down into this now, I can already see this sort of cloudy. There you go. Let me see if I can hold mine up.
Yep.
Yeah, right. That cloud is the DNA. So at the very bottom is the solution with the banana, and then that cloud in the middle is quite a bit of DNA. So I'm going to grab my vodka. And crack this bad boy open and pour a little ice cold Polish vodka across the top here. This one, oh, it's a little harder to see. Yeah, there you go. So that's actually one of the really cool things about doing the shot ones is you get a lot of this up and down. I don't know why it tends to do that, but it tends to sort of stratify. And so, yeah, that worked really beautifully. So hold your shot glass up again.
Yeah.
Yeah, you got some very DNA-rich saliva there. Okay, so we're going to go back, and the DNA in my banana thing has formed sort of a disc. And I'm going to just try to pick this up. And there you go. That's banana DNA right there.